Welcome to Peptide-Proof, your source for rigorous, independent, evidence-based peptide intelligence. Today we're looking at how Palmitoyl Tripeptide-1 signals skin to build collagen. To understand why this specific peptide is gaining so much traction in the formulation world, we first have to tackle a fundamental flaw in traditional peptide application.
The biggest challenge we face with peptides is really just a matter of compatibility. Think about the bare GHK peptide. It's a naturally occurring tripeptide with fantastic properties, but it is fundamentally a water-loving, hydrophilic molecule. Now compare that to your skin barrier, the stratum corneum. This barrier acts like a lipid-rich mortar between your skin cells, and naturally, it repels water. Because these two things don't mix, a naked GHK peptide essentially just sits right on top of the skin. If the peptide cannot penetrate that lipid layer to reach the living cells underneath, it simply can't do its job, no matter how elegant its biological mechanism might be.
And this brilliantly illustrates exactly where Palmitoyl Tripeptide-1, often just called PT1, steps in as the solution. PT1 is a synthetic peptide that takes that classic GHK sequence and simply attaches a 16-carbon fatty acid tail, known as a palmitoyl group. So why does that one little modification matter so much? Because palmitic acid is actually one of the most common fatty acids already found in human skin. By attaching it, the entire molecule transforms into an amphiphilic structure, meaning it now has a water-loving head and a lipid-loving tail. This solves the penetration problem completely. It allows the peptide to effortlessly slip right through those lipid bilayers and reach the living fibroblasts deep in the dermal epidermal junction, all without needing to rely on a copper molecule to get there.
Okay, let's break down exactly how this mechanism works, because this is where the science gets really fascinating. It relies on what's known as a matrikine biological cascade. Unlike something like copper peptides, which act a bit like a sledgehammer for broad tissue repair, PT1 is a highly precision tool. First, it perfectly mimics a matrikine, which is essentially a tiny protein fragment that your body naturally releases when your skin's collagen breaks down.
Beyond the headlines.
Into the research. Peptide-proof.
Next, your fibroblast receptors detect this fake damage signal. And finally, that specific detection triggers the TGF-beta pathway inside the cell. It forcefully shifts the fibroblast from a resting state directly into an active collagen-building state, churning out brand new extracellular matrix components.
And the clinical data we have backing up this mechanism is striking. A 2024 clinical study measuring the effects of an eye cream containing PT1 showed a 55% jump in collagen density over just 12 weeks as measured directly by ultrasound. Now, to keep things objective, we always have to look at the formulation context. That 55% increase wasn't from PT1 acting entirely on its own. It was part of a multi-ingredient formula. However, the in vitro arm of that exact same study confirmed that the peptide complex directly stimulated the fibroblast to produce collagen. So the biological mechanism is absolutely verified, and the clinical translation is clearly there when it's formulated correctly.
So, knowing that we have this highly penetrable, highly effective precision signal, the crucial question becomes, when is the optimal time to actually apply it to get these kinds of results? To answer this, we looked at some really compelling 2026 findings from the Journal of Cosmetic Dermatology that explored the internal circadian clock of our skin fibroblasts.
The research reveals a fascinating insight into application timing. They discovered that collagen metabolism strictly follows a biological rhythm. While the genes responsible for assembling collagen peak during the daytime, the genes responsible for actually synthesizing and secreting it peak at night. Because of this, a nighttime application of PT1 aligns perfectly with your skin's natural clock. This chronomodulated approach ensures that when you deliver that fake damage signal, the cell is already biologically primed and ready to ramp up production.
So how do you actually integrate this into your own regimen? Here are your practical takeaways. First, as we just saw, apply PT1 during your nighttime routine to leverage that biological peak. Second, it pairs brilliantly with other signal peptides or neurotransmitter-inhibiting peptides, like arginine, since they operate on completely different pathways without conflicting. But third, and very importantly, avoid mixing it directly with strong acids, like low-pH alpha-hydroxy acids, in the exact same step. Why? Because those low-pH environments can actually protonate the peptide, essentially altering its chemical structure and ruining its ability to bind to those fibroblast receptors. Keep them separated by time of day to give the peptide a clean, stable environment to do its signaling work. That's all for this episode. Like and comment to receive materials from us. Thanks for watching, and we'll see you next time.